Now, within biochemistry, there are 20 amino acids that are very important to understand and know because we will see these 20 amino acids in various chemical processes. We'll see them in enzymatic reactions, in signal pathways, hormone transduction. We will see these 20 amino acids in various places, so it's important to understand them so we know what's happening. So in all amino acids, remember we have a general structure that consists of a central alpha carbon, a hydrogen, that R group, the amino group, and the carboxyl group. And also note that this part is the same for all amino acids, but this R group will be different depending on what amino acid we are talking about. And in these diagrams, the R groups are labeled in orange. So this part of all amino acids is the same. So since this part of all amino acids is the same, it's important to understand their geometry. So the central carbon, it has 4 molecules bound to it. It is tetrahedral. The amino group also has 3 molecules bound to it. It's got 3 hydrogens, 1 carbon, also tetrahedral. This carbon over here is trigonal planar because remember, we have a double bond with that oxygen. So essentially we have a carbon, oxygen, oxygen. This is what we see over here. So since we have that double bond character, that carbon does not have 4 different bonds. It has a double bond. So it is trigonal planar. It is not tetrahedral. Now, and this geometry is going to be the same for all amino acids because this part of the structure is the same for all of them. Now, what's also important to note here is chirality. And remember that glycine is the only amino acid that does not have a chiral center, because in order to have a chiral center, your central alpha carbon has to have 4 different substituents. And since glycine has 2 hydrogens and it does not have 4 different substituents, it does not exhibit chirality. And remember, chirality just means that you are able to produce the molecule as a mirror image, as an enantiomer, as a non-superimposable mirror image. Now, moving forward to alanine. Alanine is the same as glycine, but it has that methyl group instead of that hydrogen as its R group. Now, since alanine has 4 different substituents, alanine will exhibit chirality, and chirality will be exhibited in all amino acids here on forward. Glycine is the only one that does not have chirality. Now, it's also important to note of these amino acids is that they are nonpolar and their R groups are aliphatic. Aliphatic is just another way of saying nonpolar. You have carbon-hydrogen bonds. Now, we know that whenever we have nonpolar R groups, if we have carbon-hydrogen bonds, they can interact with one another via the hydrophobic effect in van der Waal interactions. So if I placed a mixture of leucine into a polar solution, the hydrophobic regions of leucine will cluster together and the hydrophilic region of all the leucines will be on the outside. That's the hydrophobic effect. The van der Waal interactions are when 2 nonpolar molecules come in the close vicinity of one another due to their electrons spinning rapidly around their nucleus, creating permanent or transient dipoles. So these 7 amino acids are able to interact via hydrophobic effect in van der Waal interactions. And remember, they are nonpolar. They are nonpolar because we have these carbon-hydrogen bonds. Aliphatic just means carbon-hydrogen bonds. Now also note that these R groups are non-ionizable. They do not have the ability to donate or accept protons. Now, when we talk about the general structure of an amino acid, that carboxyl group is able to donate or accept protons, and the amino group is also able to donate or accept protons. So these 2 groups are ionizable. Now, but the R groups, in this case, they are not ionizable because we do not have any electronegative atoms. We don't have any charges. They're all nonpolar. They have no charges, so they will not have a pKa. Because in order to have a pKa, you must have some sort of an ionizable group. But since these do not have an ionizable group, they will not have a pKa. Now, proline is a little different because it has that general amino structure. So this would be the amino bond. This would be the carboxyl bond. But what we see is that the R group, it branches and then it connects with that amino group. So we get NH2 instead of NH3, and that's very important to note. So this creates a rigid conformation. So any proteins that have proline will have more of a rigid conformation because we have this carbon from the R group binding to that nitrogen. And then we have valine, leucine, and isoleucine. Leucine and isoleucine are isomers of one another. So the position of one of the methyl groups has been switched. So remember that isomer just means that the chemical structure is the same. And not the chemical structure, the chemical formula is the same, but the chemical structure is different. So essentially the same molecule, just different orientations. Leucine and isoleucine are more bulky, so they're able to interact more via the hydrophobic effect, whereas glycine, it's not really that hydrophobic because that R group is so small. But leucine and isoleucine, as you get more bulky, you're able to participate more via the hydrophobic effect or van der Waal interactions. Now, methionine, the sulfur, it actually has a electronegativity similar to carbon. So this methionine is also slightly nonpolar. And these are all the nonpolar aliphatic R groups. Now, when we talk about the aromatic R groups, we have 3 of them: phenylalanine, tyrosine, tryptophan. So this phenylalanine is relatively nonpolar and hydrophobic because if you take a look at this molecule, we do not have any charges in the R group, and we do not have any highly electronegative molecules in the R group. Tyrosine is the first amino acid that we see that has a pKa. Why does it have a pKa? Because it has an ionizable group. Oxygen and hydrogen have a electronegativity difference of greater than 0.4. So this oxygen is able to donate or accept a hydrogen because that oxygen has that high electronegativity, and the pKa is 10.1. Tyrosine is also slightly polar because of this high electronegativity and this hydroxyl group over here. Then we have tryptophan. Tryptophan is slightly polar because we can see we have this nitrogen over here. Nitrogen, hydrogen have a electronegativity difference of greater than 0.4 because nitrogen has a high electronegativity. So this will also be slightly polar. This, however, does not have any ionizable groups, ionizable molecules or atoms in its R group. So for the aromatics, there's only one ionizable amino acid, and that is tyrosine because of this hydroxyl. In our polar uncharged groups, so they are polar and they're uncharged. Uncharged is important because if we take a look at the R groups, there is no charge. And out of the polar uncharged groups, there are 3 that are ionizable. So 3 of them have highly electronegative atoms that are able to donate or accept protons. And since they have ionizable groups, they all have a pKa. So for serine, hydroxyl, threonine, hydroxyl, and for cysteine, that sulfur hydrogen bond. Now, asparaginine and glutamine, they are polar because we have those electronegative hydrophilic atoms, but they do not have ionizable groups. Therefore they do not have pKas. And when we talk about the polarity in serine, the OH contributes to polarity, same with threonine, and for cysteine, it's that sulfur hydrogen bond. Now, for the positively charged R groups. Now, what's important to note is that the most hydrophilic amino acids are the ones that are charged. It doesn't matter if they're negatively charged or positively charged. As long as they're charged, they will be the most hydrophilic. So we have 3 that are positively charged at a pH of 7, lysine, arginine, and histidine. All 3 of them have ionizable groups. So for lysine, it's that NH3. For arginine, it's that NH2. And for histidine, it's that nitrogen double bound to a carbon. Now for arginine, it is important to note that this double bond character is delocalized. So sometimes we may see that double bond here, sometimes we may see it here, but since it is delocalized, it doesn't really matter. This just means that that double bond can exist in both locations. Now, histidine is very important in our biological systems because it has a pKa of 6, which is close to the pH of 7, and pH of 7 is what we see in our biological systems. So histidine is often found in our biological systems because it has that pKa of 6. And if we recall our knowledge from buffers, we can remember that if we have a solution and we need a buffer, our buffer should be within 1 pH value from its pKa. So if we have our biological systems with a pH of 7, we should choose a buffer which has a pKa of plus or minus 7. So that's what makes histidine a good buffer in our biological systems. Lastly, we have the negatively charged R groups. Once again, they are charged, so they will be very hydrophilic. So we have aspartate and glutamate. Both of them have ionizable groups, and then we see the carboxyl groups, and since they both have ionizable groups, once again, they both have pKa's. So this is just a general overview of the amino acids. It's important to note which ones are nonpolar, which ones are aliphatic, aromatic, which ones are polar uncharged, and which ones are charged but hydrophilic. It's also important to understand which groups are ionizable and which ones are not ionizable, and to understand the relative pKa's.